Science Quest, Episode 7 Unlocking the Universe, Fusion Energy and the Quest for Clean Power. Have you ever dreamed of a power source so clean, so abundant it could solve our energy crisis overnight? Imagine a sun on Earth, a star in a bottle, providing limitless, pollution-free energy. That dream is closer to reality than you think. Welcome back to Science Quest, where we explore the cutting edge of scientific discovery. Today, we dive into the fascinating world of fusion energy, a breakthrough that could rewrite the future of our planet. Unlike fission, which splits atoms, fusion combines them. Think of it like this. Imagine two tiny bouncy balls, isotopes of hydrogen, specifically deuterium and tritium. When you slam them together with incredible force, they fuse, forming a heavier element, helium, and releasing a tremendous amount of energy in the process. This is the same process that powers our sun and other stars. It's the ultimate clean energy source, leaving behind only harmless helium. But recreating this stellar process on Earth is incredibly challenging. The sun achieves fusion through immense gravity and pressure. We need to replicate these conditions artificially, which requires temperatures exceeding 100 million degrees Celsius hotter than the sun's core. The International Thermonuclear Experimental Reactor ITER, a massive international collaboration involving 35 nations, is leading the charge. Located in Cadarache, France, ITER is building the world's largest tokamak, a donut-shaped device using powerful magnetic fields to confine and heat plasma, the superheated state of matter needed for fusion. Think of the magnetic field as an invisible cage, holding the superhot plasma away from the reactor walls. If the plasma touches the walls, it cools down and the fusion reaction stops. ITER aims to achieve sustained fusion reactions, proving the scientific feasibility of this technology. While still in the experimental phase, recent advancements have been incredibly promising. Scientists have achieved significant progress in plasma confinement and heating, bringing us closer to sustained fusion reactions. However, significant challenges remain. First, achieving and maintaining the required temperature and pressure is extremely difficult. Second, the materials used in the reactor must withstand extreme conditions. Third, the energy required to initiate and sustain the fusion reaction is currently higher than the energy produced, a challenge known as the energy break-even point. Overcoming this requires further technological breakthroughs. If successful, fusion energy could revolutionize our world. Imagine a future free from reliance on fossil fuels, with limitless clean energy for electricity generation, transportation, and industry. This would dramatically reduce greenhouse gas emissions and mitigate climate change. However, it's crucial to acknowledge the timeline. While ITER aims to achieve sustained fusion reactions by the mid-2030s, commercial fusion power plants are likely decades away. But the potential rewards are enormous. The quest for fusion energy is a testament to human ingenuity and our determination to create a sustainable future. Let's test your knowledge. True or false, fusion energy produces radioactive waste? Answer: false, primarily produces harmless helium. Now, what are the two isotopes of hydrogen used in fusion reactions? Answer: deuterium and tritium. Fusion energy represents a beacon of hope in our quest for a sustainable future. While the path is challenging, the potential rewards are immense. From conquering the challenges of plasma confinement to building commercially viable reactors, the journey to unlock the power of the stars is a remarkable scientific endeavor. Next time on Science Quest, we'll delve into the mind-bending world of quantum computing and its potential to revolutionize everything from medicine to artificial intelligence. Stay tuned. Science Quest, Episode 1 The CRISPR Revolution, Editing Life's Code. Ever wished you could rewrite the code of life itself? Sounds like science fiction, right? Well, hold onto your hats, because today we're diving into the incredible world of CRISPR-Cas9, a gene editing tool so powerful it's rewriting the future of medicine and beyond. We'll explore what exactly CRISPR is, its groundbreaking applications, and the ethical considerations that come with wielding such immense power. Get ready to explore the cutting edge of. Imagine a highly sophisticated molecular scissor. That's essentially what CRISPR-Cas9 is. Derived from a naturally occurring bacterial defense mechanism, CRISPR is a revolutionary gene editing technology allowing scientists to precisely target and alter specific DNA sequences. Think of your DNA as a vast instruction manual for building and operating your body. CRISPR allows us to correct errors in this manual, potentially curing genetic diseases. The Cas9 part is an enzyme that acts like the scissor, cutting the DNA at the precise location guided by a special RNA molecule, like a GPS system, guiding the scissors to the correct spot. Unlike previous gene editing methods, CRISPR is significantly more accurate, efficient, and affordable, making it accessible to a wider range of researchers and applications. This has led to an explosion of research in diverse fields. The potential applications of CRISPR are almost limitless. Let's explore some of the most promising areas. Imagine a world without cystic fibrosis, sickle cell anemia, or Huntington's disease. CRISPR offers hope for treating these debilitating conditions by correcting the faulty genes responsible. Clinical trials are already underway, showing promising results. CRISPR can be used to engineer immune cells to target and destroy cancer cells more effectively. It can also be used to correct genetic mutations that contribute to cancer development. This approach is revolutionizing cancer treatment strategies. CRISPR can be used to enhance crop yields, create disease-resistant plants, and improve nutritional content. This offers a powerful solution to address global food security challenges. Imagine drought-resistant crops thriving in arid regions or disease-resistant bananas combating devastating fungal infections. While CRISPR potential benefits are immense, we must also consider the ethical implications. The ability to alter the human germline that genes pass down to future generations raises profound questions about unintended consequences and the potential for misuse. What are the societal implications of using CRISPR to enhance human traits beyond correcting disease? Should we be allowed to create designer babies with specific physical or intellectual characteristics? These are complex questions that require careful consideration by scientists, ethicists, and policymakers. Research on CRISPR is constantly evolving. Scientists are developing more precise and efficient versions of the technology, minimizing off-target effects, accidental edits in the wrong parts of the DNA. They are also exploring new delivery methods to ensure the CRISPR system reaches its intended target effectively. Imagine targeted therapies delivered directly to specific organs or even individual cells within the body. The future of CRISPR looks incredibly bright, but responsible development and rigorous ethical guidelines are critical to ensure its benefits are realized while mitigating potential risks. CRISPR-Cas9 has undeniably revolutionized the field of gene editing, offering remarkable potential for treating diseases, improving agriculture, and furthering our understanding of biology. While ethical considerations remain at the forefront of this exciting field, the advancements made in CRISPR technology show us that the ability to rewrite the code of life is no longer science fiction fantasy, but a powerful reality we must navigate responsibly. Next time on Science Quest, we'll explore the mind-bending world of quantum computing, a technology that promises to solve problems beyond the capabilities of even the most powerful supercomputers today. Don't miss it. Science Quest, Episode 1: Unlocking the Universe, CRISPR, Gene Editing, and the Future of Life. Did you know that scientists can now edit our genetic code with the precision of a word processor? It sounds like science fiction, but it's the reality of CRISPR-Cas9, a revolutionary gene editing technology that's rewriting the future of medicine, agriculture, and even our understanding of life itself. Welcome to Science Quest, where we explore the cutting-edge advancements shaping our world. CRISPR-Cas9, short for clustered regularly into space short palindromic repeats and CRISPR-associated protein 9, is a gene editing tool derived from a natural defense mechanism found in bacteria. Imagine tiny molecular scissors that can precisely cut and paste DNA sequences. These scissors are guided by a short RNA molecule that acts like a GPS, directing the Cas9 enzyme to the exact location in the genome where the edit needs to be made. Unlike older gene editing technologies, CRISPR is remarkably precise, efficient, and relatively inexpensive. This makes it accessible to a wider range of researchers and opens up incredible possibilities across various fields. The potential of CRISPR in medicine is staggering. Imagine a future where genetic diseases like cystic fibrosis, Huntington's disease, and sickle cell anemia are eradicated. CRISPR is already showing promising results in clinical trials targeting faulty genes that cause these debilitating conditions. Researchers are also exploring its use in cancer therapy, designing immune cells to more effectively target and destroy cancer cells. But it's not just about treating existing diseases. CRISPR could revolutionize preventative medicine, allowing us to identify and correct genetic predispositions to diseases before they even manifest. With such powerful technology comes significant ethical considerations. Germline editing, modifying genes in reproductive cells that pass down to future generations, raises concerns about unforeseen consequences and the potential for unintended mutations. The possibility of designer babies, selecting for specific traits like intelligence or physical attributes, sparked intense debate about the equity, access, and societal impact. Responsible development and regulation of CRISPR technology are crucial. Open discussions involving scientists, ethicists, policymakers, and the public are essential to navigate these complex ethical challenges and ensure that CRISPR is used for the betterment of humankind. The applications of CRISPR extend far beyond human health. In agriculture, it's being used to create crops with enhanced nutritional value, increased yields, and greater resistance to pests and diseases. This has huge implications for food security, particularly in regions facing climate change and resource scarcity. Scientists are also exploring CRISPR potential in environmental remediation, developing organisms to clean up pollution and restore ecosystems. The possibilities are truly limitless. CRISPR-Cas9 is a revolutionary tool with the power to reshape the future of life. Its potential to treat diseases, enhance agriculture, and solve environmental problems is undeniable. However, ethical considerations must guide its development and application. Next time on Science Quest, we delve into the fascinating world of quantum computing, the technology that could revolutionize everything from medicine to artificial intelligence. Join us as we explore the mind-bending possibilities of a world powered by quantum mechanics. Science Quest, Unlocking the Secrets of the Universe, Episode 1, CRISPR Gene Editing, the Future of Medicine. Imagine a world where genetic diseases like cystic fibrosis, sickle cell anemia, and even cancer are eradicated. Sounds like science fiction? Think again. Today on Science Quest, we dive into the revolutionary world of CRISPR gene editing, a technology so powerful, it's rewriting the rules of biology and holding the key to a healthier future. Search terms: CRISPR gene editing, gene therapy, genetic engineering. CRISPR stands for clustered regularly interspaced short palindromic repeats. Sounds complicated, right? But the concept is surprisingly elegant. Imagine your DNA is a vast library filled with books, genes containing instructions for your body. Sometimes there are errors in these books, mutations leading to diseases. CRISPR is like a highly precise pair of molecular scissors and a copy editor combined. It's a gene editing tool derived from a natural defense mechanism in bacteria. A protein called Cas9 acts like the scissors, cutting the DNA at a specific location guided by a short RNA molecule that acts as the address label. This precision allows scientists to remove, add, or alter specific DNA sequences, potentially correcting genetic defects or modifying genes to enhance desirable traits. It's like finding a typo in a book and correcting it, but on a molecular level. The applications of CRISPR are astounding. In the fight against cancer, scientists are using CRISPR to engineer immune cells to more effectively target and destroy cancer cells. This is personalized medicine at its finest. In inherited diseases, CRISPR offers hope. Researchers are working on correcting faulty genes responsible for conditions like sickle cell anemia and cystic fibrosis, offering the potential for a cure. Even genetic disorders affecting the brain, such as Huntington's disease, are being investigated with CRISPR-based therapies. With such powerful technology comes a responsibility to address ethical concerns. The possibility of designer babies using CRISPR to enhance traits like intelligence or athleticism raises significant ethical questions. We need to carefully consider the societal implications of altering the human germline, ensuring equitable access to this technology, and preventing its misuse. The scientific community is actively engaged in establishing guidelines and regulations to ensure responsible development and application of CRISPR. Search terms: CRISPR ethics, gene editing ethics, designer babies. The future of CRISPR is bright, but it's also complex. Scientists are constantly improving its precision, efficiency, and delivery methods. We're exploring its potential in treating a wider range of diseases, from genetic disorders to infectious diseases. Moreover, its applications extend beyond human health. CRISPR is being used in agriculture to develop crops with enhanced yields and resistance to pests and diseases. It holds promise in environmental remediation, even in biofuel production. CRISPR gene editing technology is transforming our understanding of biology and medicine. Its potential to cure diseases and improve human health is immense, but careful consideration of ethical implications is crucial. We are only beginning to scratch the surface of what this technology can achieve. Next time on Science Quest, we'll explore the exciting breakthroughs happening in the world of fusion energy, the ultimate clean energy source that could power our future. Join us then. Search terms: fusion energy, breakthrough, clean energy, renewable energy. Science Quest: Unlocking the Universe, Episode 1: The CRISPR Revolution. Imagine a world where genetic diseases like cystic fibrosis and sickle cell anemia are eradicated, where we can precisely edit our DNA to prevent inherited disorders before they even manifest. Sounds like science fiction? Think again. Welcome to Science Quest, where we explore the groundbreaking advancements shaping our future. Today, we're diving deep into the revolutionary world of CRISPR, a gene editing technology that's rewriting the rules of biology. CRISPR, short for costed regularly interspaced short palindromic repeats, isn't some alien technology. It's a natural defense mechanism found in bacteria. Bacteria use CRISPR to remember and defend against viruses. Scientists cleverly adapted this system, pairing it with the Cas9 enzyme, a molecular scissor that can cut DNA at specific locations. Think of it like a highly advanced find and replace function for our genetic code. This precision is key. Unlike previous gene editing methods, CRISPR is significantly more accurate and efficient, allowing scientists to target specific genes within the vast human genome with remarkable accuracy. This means we can potentially correct genetic defects responsible for a wide range of diseases. Imagine eliminating the genetic predisposition for certain cancers or Alzheimer's. What ethical considerations do you think we need to address when discussing the potential of gene editing? Let us know in the comments below. While disease treatment is the most prominent application, CRISPR potential stretches far beyond the realm of medicine. Imagine disease-resistant crops requiring less pesticides and herbicides, leading to more sustainable and efficient farming practices. CRISPR is already being used to develop crops with improved yields, nutritional value, and resilience to climate change. Engineered algae using CRISPR could become highly efficient biofuel producers, offering a sustainable alternative to fossil fuels. This could dramatically reduce our carbon footprint. CRISPR could help revive endangered species by correcting harmful genetic mutations or by making them more resistant to diseases. Despite its incredible potential, CRISPR technology isn't without its challenges. Off-target effects, unintended cuts in the genome are a major concern. Researchers are constantly working to improve the accuracy and specificity of CRISPR systems. More significantly, the ethical implications of gene editing are profound. Germline editing, modifying genes in reproductive cells, raises concerns about unforeseen consequences for future generations. Should we be editing the human germline to enhance traits, and who decides which traits are desirable? These are complex questions that require careful consideration and open public discussion. Despite the challenges, the future of CRISPR looks incredibly bright. Scientists are continually refining the technology, developing new CRISPR systems with improved accuracy and exploring new applications. We might soon see CRISPR-based therapies becoming routinely used to treat a wide range of diseases, leading to a healthier and longer lifespan for millions. Take our quick quiz on CRISPR. Test your knowledge on this revolutionary technology. Link to online quiz. From combating inherited diseases to revolutionizing agriculture and conservation, CRISPR is changing the world in ways we are only beginning to understand. Its power and potential come with significant ethical responsibilities, demanding careful consideration and open dialogue. Next time on Science Quest, we'll explore the incredible advancements in fusion energy, the quest for a clean, virtually limitless power source. Will we finally unlock the power of the stars? Find out next time.